In this project, we fabricate electrical chip, and then we also create a microfluidic chip where we can create a natural biomimetic environment for the cell to survive. And then we assemble these two chips together where we can simulate and observe the living cell behavior and then get biological information on them. So as the primary goal of this project is to understand the reaction, the protein generation in the cell, in a living cell, under stress conditions, we use a genetically modified cell line that is transfected with green fluorescent protein so that we can observe the change, the protein expression in the cell using a fluorescent microscope. Here I have the opportunity to work really closely with both the biologists and go to the lab, use my engineering skills and background, fabricate the device, and then test it. So since it's a very close family of laboratories that work very closely with each other, I find it as a researcher a very nice opportunity to bring my engineering knowledge into hands-on experiments.
A neuron is the only cells who could create a bioelectrical signal. So my project is to interface neuron with electrical chip for recording neuronal activity. A neuron, it's a cell with a body and arm. So when you let cells in a culture, neurons go on the substrate and create arms named neurites. And I would like, in my topic, guide this neurite and position in the right position the cells. So as you could see on my screen, it's the problematic of my research. Because here I create some micropillars to record neuronal activity. But the problem when you deposit cells on the substrate, neurons go everywhere and not only on the micropillars, as you could see, but grow everywhere. So most of the time we have a sensor without any neuron on it. So these pictures represent my problematic. I would like guide neuron on my sensor, that means on my micropillar arrays. So this is a model in 2 dimensions, but currently I'm also working in a 3-dimensional model and to be really close to the brain. So here we could see neuron cells are positioned on micropillars array and neurites are guided between every micropillars arrays. So we have created a neuronal network. So this collaboration helped me to have 2 really strong topics and to combine both. That means I have all the knowledge for sensor in Germany and all the biohybrid technology here in Japan. So it's really important for me to have this collaboration to mix both and to create new topics.
It happened when I was doing my master's. My supervisor told me that there's a project in Japan, if I wanted to go. And I wanted to extend my vision and gain some different perspective. So I thought, yeah, I should come to Japan and learn some new experiments, new technologies, gain some new perspective. So that's why I'm here, yeah. And also there's a huge scientific community here that stimulates the research and science even more. So yeah, this is already quite hardened. What I'm working on is application of liquid TEM-cell. This is a small device that we can encapsulate liquid, and along with liquid we can encapsulate some biological specimen so that we can visualize them in their natural environment under electron microscope. So electron microscope has a vacuum chamber which doesn't allow natural visualization of biological samples inside the microscope because they are not used to stay in the vacuum, let's say. So by using this device, we want to visualize image biological structures like viruses, liposomes, bacteria even. That's what we are trying to do. So our device contains a very thin membrane and it's electron transparent. So with the electron beam, we can penetrate through the channel, and inside the liquid we can visualize these biological structures.
So I like to make nanoparticles, which can encapsulate pharmaceuticals and help to carry these pharmaceuticals throughout the body and deliver them to where they are needed and to keep them away from places where they are not needed, so they don't do any damage to healthy tissue. So what I'm starting out with is to study the material in terms of mechanics and electrical behavior, so that I can get a good understanding of how to fabricate these particles. So here you can see the membrane of the thin film that's formed on the interface between two water droplets in oil. It's a nice opportunity to be here at the prestigious University of Tokyo, also to be in Japan, which I never thought I would visit professionally. I think it's a very valuable experience to see how another lab works and what technology they use, because you can be in the same field while still using other fabrication methods, for instance, and certain traditions grow, and it's very important, I think, to learn from these traditions and established operating procedures from another lab, because there's a lot of effort that goes into developing this.
The topic of this project is advanced microsystem based on the biodegradable materials or the dissolvable materials. So here is the bombyx cocoons. Then we use these materials to get the silk film. Here you see, this is the transparent silk film, and this film is biodegradable. If you put this film in the water, so it disappeared completely. We don't do the research to the fabrication process of these materials, but we wish to use the micro-nano fabrication technique to get some functional materials based on these materials. For example, for some smart drug delivery system and also micro-energy harvesting. The final target is to realize some, for example, the bio-MEMS device or system. So based on these biodegradable materials. And for example, some implanted applications inside body, but is very safe to the human. So that is the target of this project. And also, for example, the micro-energy harvesting, we wish to use these materials to realize some micro-energy harvester, which, for example, as an attractive vision of the future, we wish to realize some implanted device also inside body to harvest the body motion energy and convert it to the electrical power. So I think it's a very nice platform for the young researcher to do the very attractive work here.